the mysterious and sophisticated human brain. It determines the way we think, feel, and act.
We kind of figure out many general fundamental things about the brain, but we're still very far away from understanding how the brain generates our feelings, our thoughts, and exactly how the brain encodes every information in our brain, like memories.
There are an average of 8.6 billion neurons connecting different parts of a brain. That's a billion with a B. They weave an immensely complex and interconnected network inside our skulls.
Neurons and neuronal connections are only on a nanoscale in size. Our brain computes at millisecond time scale, probably faster than any other physiological process we study on cellular level.
Space and time. These two major factors have been preventing neuroscientists from better understanding our brains, but efforts have never ceased. Microscopes reveal minuscule wonders by making things seem bigger. Though such optical instruments have kept improving in the past 400 years, there is always limitations for resolution and cost. That creates a problem in studying objects at a nanoscale, like neurons, the primary functional unit in the brain.
And we decided to go in opposite direction. How about instead of magnifying the image of the object, we magnify the object itself. We blew it up.
Blowing the object up, the idea itself is so crazy that even Dr. Piakavich himself did not believe it was possible. Until Dr. Ed Boyden's team at MIT, where Kirill Piakavich was working at the time, came across a type of self-expandable hydrogel, which is essentially made from a type of water-absorbing polymers. Wait, is its function the same as?
So it's the same hydrogel molecule used in baby diapers.
The new substance can soak up water many times its own weight. During this process, each hydrogel polymer chain expands equally, allowing the materials to grow exponentially in size, up to 1,000 times bigger. And how this tool can help us better observe our brain.
I remember an analogy from my childhood when I was writing something on a balloon and I was blowing up the balloon, and I see how the small writings on the balloon were blowing up, and I could see the details, fine details inside.
Baby diaper chemistry or childhood games may offer new inspiration for scientists for a better view of our brain's wiring. But practice is another story. To allow this idea works, Dr. Piakavich embeds a slice of brain tissue into the hydrogel. The gel is then fully immersed in water. On the nanoscale, the molecules of the brain tissue slice stretch with the hydrogel molecules attached to them. You can even see the expansion with the naked eye. This method is known as expansion microscopy. Though the gel itself can grow equally in all directions, how do we ensure the sample tissue inside the gel will expand proportionally to keep its structure from being distorted?
This is actually a very good question, because we don't want to image artifacts of expansion, we would rather image the structures itself. So major reason why the expansion seems to be fully isotropic or equal in all direction is because of the hydrogel monomers that we infuse, they only less than nanometer in size. And we ensure that these monomers evenly cover the biomolecules around them.
In other words, the nanometer scale hydrogel monomers are like tiny little hooks, ensuring that there is enough hooks that distributed as evenly as possible. They tightly grab onto each molecules of the brain tissue, then pull them towards their own growth directions without distorting the original shape. And most importantly, into a much larger size. With this technique, which has been further developed in Dr. Piakavich's lab at Westlake University, a sample slice can expand up to 8 times in length, or roughly 700 times in volume.
Because of this technology is so simple, so cheap and robust, it's spreading all over the biological sciences. Right now in the United States, there are trials for expanding the clinical specimen to get a super resolution imaging of some human tissue to diagnose early stages of certain diseases which are not visible with a conventional microscope.